Hello everyone. In this video, we will discuss how to use iDEP tool. In our last video where we discussed the paper review of iDEP along with what methodology they used, how they make this tool and how this tool is different from other tools. So if you have not watched my previous video, you can just search it, or either you can go to my YouTube channel where you can search it by Qurat-ul-ain Bioinformatics. This is basically the part 1 where I had discussed each and everything about the paper review, about the paper of iDEP. And so if you have any confusion or any problem like using this tool, you can just go to their paper and can see. So let's get started and understand how this tool works. This is basically the link that is made by South Dakota State University. This is the home page of iDEP tool, and here it shows that by default Homo sapiens species has been selected. So here you can go to this select tab and click any of the button. Like if you want to select mouse genes, we'll just click on it and click over there, and now it has selected this mouse species. But I want to work on human genes, so I will click on it, and then if you want to customize these species, you can just upload the custom pathway.gmt file. Now here we will work on this demo dataset, so we will not upload our file over there, but you can upload your file in CSV format, text format, or Excel format. Here the data types on which you can work is read counts data that is recommended, or if you have your normalized expression data, means your data is already normalized, iDEP will not normalize it. And next is fold changes and adjusted p-values. If your file contain fold changes and adjusted p-values, you can just select it and can drop your file over there. Now here two groups are selected in humans. And you can also upload your expression design file in CSV or text form. So let's click on this demo button because we want to choose the demo dataset in this video. These are the sample gene IDs, and these are the samples that are total 6 in numbers, and these values shows the differential expression rate of each of these ensemble gene IDs. If you want to find out the gene symbols, you can just copy this gene ID and can go to the ensemble and can check its gene symbol. So our next step is to perform pre-processing on it. So the minimum count per million rate is by default it has been set to 0.5, and number of libraries are set to 1, and the method used for clustering and PCA is HR, and pseudo count is made equal to 4, and missing values will use gene median. Now here you can use all of these buttons to download your basically processed data, converted counts, etc. This image shows the bar plot where this red color shows the control samples and this blue color shows the treatment samples. You can just click on this button and can download your image in the form of PDF, PNG, or SVG. So let's click it on PDF button and download it. Next image that has been generated is the box plot. Now this box plot shows that these dots are the outliers, and this shows basically the spread rate of our dataset, and this central line is the median line. If our data is normalized, then this line is completely in center. So this means our data is completely normalized. There is no need to further normalize it. Next is the density plot. Then we have the scatter plot, where most of the expression values or differential expression lies in this range. This is the dispersion graph, but don't worry, we will discuss all of these findings in our next video. In this video, we will just create these images and we'll learn how to use this tool. In our next part, we will basically discuss what these colors shows, what this blue line shows, and same goes for all of these plots. So this is the QC, basically represents the quality control. Now it shows that what is the quality of our expression data. So some of the genes are coding genes. These are the pseudo genes. These are long non-coding RNA, and these are the unknown genes whose function is not known. And this is the artifact. So it shows the number of genes. So most of our genes, like 19,304 genes, are coding genes. 87 genes are pseudo genes, and same goes for long non-coding RNA. These are 87. 16 are genes are unknown, and 12 are artifact. Next is the gene plot that will shows its axis is 45, and it will shows the expression level of normalized expression. Now these are some of the outliers, and it shows the rate of expression. So these are the AREG and PTGS2 genes. These two genes has been selected to show their expression rate. You can also select some more genes that are present in your data, like let's select this gene, and now it will show the comparison of these three genes. This is the expression rate of our gene in control samples, and now its expression has been increased in HOXA1 knockdown, and same goes for this gene MAGIX and PTGS2. All of these three genes expression has been increased in HOXA1 knockdown. So this means that this problem has been arise because of the higher level of expression of these genes. Click on this data file, and now here previously that I had showed you, like you can just copy these gene IDs and can go to the ensemble. So now there is no need to go to the file and to find out their gene symbols. So these symbols basically represent all of the gene symbols. Is the information button where it shows that how we had basically performed the pre-processing. So first of all, they filter out genes with extremely low expression rate, and secondly, they convert gene IDs to ensemble gene IDs or string DB gene IDs, and third is they apply transformations. Now here in the filtering button, they had explained like some genes are not expressed in any samples, others are expressed at extremely low levels, and we need to remove these genes from further analysis. So by default, a gene must have more than 0.5 counts per billion in at least one sample, otherwise the gene is removed. Users can specify that genes must be above 0.5 cpm in two samples by changing the number of libraries option to 2. Now it shows that the number of libraries means like the samples count per million must be present in at least one sample. So this number of libraries shows the number of samples, like the gene that you are going to remove or you are going to be added into your data. It must be expressed in at least one sample. And then we have CPMs are calculated by normalizing the read counts by the total counts per sample. If sample 1 has 20 million counts in total, then the read counts are divided by 20 to get the CPM. So in this way, they had shows like how they control the CPM, how they apply the transformation, and here are the 3 buttons for transformation. Basically, read all of these and for your understanding. Now here this tab basically represents that we have 19,000 almost 19,000 genes in 6 samples, and from this 19,000 genes, 13,819 genes have been passed from filtered, and 13,729 genes are and almost 13,729 genes have ensemble gene IDs in our database. So remaining 90 genes were kept in the data, means remaining 90 genes has been removed or kept in the data where their ensemble gene IDs has not been found in the ensemble database. Now let's move toward the clustering part where we will see how to generate a heat map, how to perform the standard deviation distribution, and how to generate the sample trees. So it may take some time to create heat maps, so be patient and not close this window. Now here on the on this side, it basically shows that for performing clustering, basically there are 2 options. First of all, we have hierarchical clustering and there is k-mean clustering, and the number of genes that they will shown are the 2000 genes. And for calculating distance, they use Pearson correlation matrix. Other options are Euclidean distance, absolute Pearson, etc. Then we have the linkage, it will shows how much linkage will be present in these genes to show them in the clustering. So the linkage that they have selected by default is average. Other options are complete, single, median, centroid, or MCQT. Sample colors has been set to by default to names. According to their name, you will color them, and Now here this heat map has been generated that shows two types of samples. This is red is the control sample, blue is the treatment sample, and now it clearly shows that the genes that are present in control samples have blue expression that is shown by green color, but the genes and or now when they move towards the treatment samples, their expression has been highly increased and now it becomes red. And now this basically this scale basically also shows the level of expression rate, and same goes for these genes. Now in the control samples, these genes have high expression, but in treatment samples their expression has been reduced, and now they are represented by green color. So now here this shows that you can click on any region to zoom in and you can see that region. So let's select this area to see the heat map in detail. Now it is creating the sub heat map from this primary heat map. Here it shows the sub heat map. These are basically the gene symbols for which these expressions have been represented. This is basically the whole area for which the heat map is represented. This is the control 2, control 3, Hoxa 1, 2 and 3. So basically it shows the heat map in 5 samples. It has removed the control 1 sample. Here you can see the enrichment tree, where you will see like in which pathways most of our genes are present. So these are the pathways in which most of your genes are present, and it will shows the tree like in which pathways your genes are present in the form of a tree. Here you can see now here in the form of a network, you will see how many genes are present. Now this is the plot that shows like most of our genes are present in cell cycle pathway, then cell adhesion molecules, some are Fanconi anemia pathways, some are involved in homologous recombination, some are involved in DNA replication, some are involved in biosynthesis of amino acid, and some are in oocyte meiosis. Now here you can change these parameters from these, and you can check what is the effect of it on changing them. Now it shows basically all of the gene. These are the ensemble IDs. Now it has converted these ensemble IDs to entrance gene IDs, and also shows them like on which chromosome number these genes are present, along with little description like what is the role of these genes. Here the in detail section, they basically represented like how they perform enrichment. This is the standard deviations of all genes, where most of the genes expression is upper 2000. This is the sample tree where control 2 and 3 expression is highly similar with each other. Then they have expression with control 1. Now here Hoxa knockdown 3 and 1 has similar basically expression rate. Now to calculate this distance, they choose Pearson correlation along with and the linkage that they had selected as average linkage. Now let's move towards principal component analysis, or PCA. For PC2, the amount of variance is 5.5%, and for PC1, it is 81.7%. So it clearly shows that how much there is variance in both of these samples. These squares represent the PC1 or the control samples, and blue color shows the treatment samples or Hoxa 1 knockdown samples. This is the 3D structure of PCA, where it shows on this side, these are basically the control samples, and these are the treatment samples. Now let's move towards PCA tools that has been used for generating these diagrams. So this is the principal component score. These are basically the scores from -20 to 20, and here -30 to 30. These are the scree plots that shows elbow plots and horns plot, contribution of genes to PC1. So there is highest contribution of this gene AREG, that is 0.64%, and lowest percentage is 0.23%. Now this is the contribution of genes to PC2. It was about PC1 and it is about PC2, and highest contribution is 1.93% of this gene, and lowest contribution is of this gene whose gene symbol is not known. Now let's move towards differential expression analysis. The method that they had used for this analysis for differential expression, they use DSeq2. You can also use Lima-Voom or Lima-Trend. Now here the FDR or the false discovery rate cutoff they had selected is 0.1 and minimum fold change is equal to 2. And then click on this submit button to run this DEG analysis. Now this analysis is being running and it shows this 971 genes that are downregulated, and this red color shows that the genes that are upregulated, these are about 1176 genes. And it shows the comparison between control and HOXA1 knockdown. Now this is the Venn diagram and upset plots. Now let's click on this submit button, and it shows that in the genes that are upregulated in HOXA1 knockdown versus control, there are about 971 genes that are basically upregulated, and 1176 genes are downregulated, and total genes are 11,672. This is the R code that they has used for this analysis. You can so here from here you can just copy it and can run it in RStudio. This is the heat map that we had seen earlier. Now, if you want to display all DEGs, you can click on this button. But I want to show only 40 genes that are displayed. So I will click on this button and I will sort them by the fold change value. And the comparison I had used is this control sample. So this is basically the heat map that has been generated. Now I can click on some of the small region to zoom in. This is the volcano plot. This is the MA plot, scatter plot. Click on this region. This shows the scatter plot, and this is the enrichment file that shows like how many genes are upregulated along with their adjusted p-value, fold change, and pathways. You can also click on this pathway and you may go to in detail of these pathways. And it is has been basically linked to KEGG pathway. Now here you can see the pathway in detail. It shows all of these pathways in the form of a tree, like those that are highly linked with each other are just shown by these sister lines. Now to generate interaction network, they use StringDB. So let's click on this button, network, and to see the interaction between these genes and between these pathways. These are all of the genes that we had submitted earlier, and total downregulated genes are 1171. Now next move towards the finding significant pathways. Oh, here it shows like which of the pathways are downregulated and which pathways are upregulated, along with the genes, along with the amount of genes involved in each pathway and adjusted p-values. Now let's click on this tree button. This is the tree that we had seen earlier. This is basically the network diagram. This is here. This is the network. Now here, these three pathways are highly interconnected: mismatch, repair, nucleotide excision, repair, and DNA replication. Now here, these two pathways are interconnected with each other, and in this way you can basically zoom in and out and to see it in detail. And it shows what are the methods that they have used to perform this analysis. Now let's move toward the genome part, where we'll see how they basically represents these chromosome characters. Now it shows like how many genes are present in on chromosome X, chromosome 22, 21, 20, and so on. And most of the genes are present on chromosome number 1, along with the positions. Now it shows like which genes are highly connected with each other, and this is the network plot. And now next move towards the module plot. Okay, now this is the about page of the iDEP, where they had explained like how who made this tool, and this is the iDEP paper. This is here is the GitHub link and database link. These are the previous versions of iDEP. It shows the privacy policy, and for contacting you can use these emails. And now here they had explained like they had no guarantee of accuracy, like they had a small team with limited resources, and we have not thoroughly tested it. So please verify all findings using other tools or ourselves. Don't completely rely on this tool, because it has not very much accuracy. These are the basically bugs that they had found in 2024, and these are the R section info, where they had explained all of the packages that they had used for R programming language. So that's it for this video. If you have any question, drop down your question in the comment section, and share this playlist with your friends so they can also get benefit from it. So that's it for this video. See you in the next video.